Hello, everyone. First, I would like to thank Twist Bioscience for inviting me to this conference. I'm Astrid Musnier. I'm the head of biology in Mapsilico. So what we aim at doing in Mapsilico is to provide computationally designed antibody drugs using AI and deep learning-based algorithms. So a little bit of history. So we were founded in 2017 from academic labs in France. And today we are 15 employees. We are a pluridisciplinary team composed of computational scientists, biologic researchers, and software developers. On the right side of the screen, you can see the executive team. And actually, Anne, Thomas, myself, and Vincent, we are the co-founders of the company. So the context of our research is antibody discovery. So as you may know, antibody discovery is a funnel-shaped process that comes with a huge attrition rate. So starting from millions of antibodies, successive rounds of selection are applied in order to identify the 1 to 5 candidates that can enter the clinical trials. And if the campaign is successful, maybe one of these molecules is going to reach the market at the end. So the whole process takes approximately 10 years and costs millions of euros. So what we aim at doing in Mapsilico is to rationalize this process in order to optimize, de-risk, and speed up the discovery phase. So in order to do so, we defined 12 tools solving the major pain points of antibody discovery. I'm not going to detail all of them, but for example, we have a tool for epitope mapping, which is called MAbTope, and it's the one I'm going to detail after in the example I'm going to show you. We have also a tool for epitope binning. We have a tool for humanization, which is MAbHuman. We have a tool for affinity maturation, for example, which is MAbMature. So all these tools have equivalents in wet lab, but we have also tools that don't have any equivalent in the wet lab. So for example, MAbSubstitute, which is a tool that aims at identifying new antibodies, starting from an initial one. And this new antibody will have a different sequence, but will still display the same effect. So one thing that is important, that using AI allows to change the process completely. Meaning that the conventional way to identify antibody is very linear because first the antibodies are selected, then they're characterized, and then they're optimized. And this is dictated mainly by the number of molecules that can be processed at each of these stages. Actually, using a computational approach, the number of molecules is not important anymore because we can optimize a huge number of sequences. So actually, the process can be changed completely by an iterative and circular process that you can see here. And we hope that defining and developing this process will allow to go from the target to the final antibody leads in just a few days. So our process, importantly, only require to have the sequence of the antibody to start. So that's something that we are really attentive to. All our tools can be used without any biological input. The result can be refined if there are biological evidence behind, but to work strictly, they just need the antibody sequence. So meaning that we can start as soon as the antibody is sequenced. So even at the antibody bank stage, for example. We estimate that this computational approach can remove the risks because very soon you can identify the best leads and exclude the bad leads, and that the acceleration factor will be around 500. It will also help to expand the IP protection because the antibodies are better characterized. So all our artificial intelligence-based tools rely on a toolbox composed of 4 databases of antibody structures, sequences, antibodies of known targets, and antibodies of known KDs. and on 4 algorithms. So a docking algorithm, which is used for the epitope mapping, a similarity algorithm, an affinity prediction algorithm, and a productivity prediction algorithm. The last one is actually in R&D right now. So I'm going to present now a case study about an epitope mapping. So it was a collaboration with Wichita State University, and the purpose was to identify the epitope of a GlycoForm-specific anti-FSH antibody. So first, a little point about FSH. So it's a pituitary hormone which plays a central role in reproduction. And it's actually the hormone that is used in clinic to induce spermatogenesis and follicular development during reproductive therapies. FSH is dimeric, so it's composed of an alpha subunit that is common to all the glycoprotein hormones, and a beta subunit that is specific to each of them. The FSH triggers its effect through a GPCR that is located on the testicular Sertoli cells and the ovarian granulosa cells. So as I mentioned in the slide before, the FSH belongs to the family of the glycoprotein hormones. So it means that it's glycosylated. Actually, FSH is glycosylated on both subunits, alpha and beta. But what's going to interest us right now, it's the glycosylations on the beta subunit. So actually, there are 2 major sites of glycosylation on the beta subunit, bound on the N7 and the N24. And 3 major forms of FSH were identified. An FSH of 24 kilodalton, so which is called the FSH24, which is glycosylated on both, and FSH21 and FSH18, which are glycosylated on one or the other. What's interesting here is that these glycosylations isoform display different effects on the cells. So the lighter forms, the FSH21 and 18, bind faster to the receptors, as you can see here on this time lapse. They also bind with a higher affinity, as you can see here by the displacement curves. And they also bind more binding sites on the cell, as you can see here on this graph. So it means that the lighter forms of FSH are more efficient. Another thing is that the FSH21 ratio decreases along the age, meaning that FSH21 is the major form in the youngs, and along age, the amount of FSH21 decreases, and the amount of FSH24, so the heavier form, increases. So it means that also these lighter forms are related to the fertility status of the woman. So what's important here is that FSH glycosylation is an indicator of the fertility of the woman and a predicator of the responsiveness to these fertility treatments. However, the glycosylation of the FSH beta is poorly estimated before being injected in the patients. So that was the purpose of the study of this Josh Bullfield group at the Wichita State University. It was to develop glycosylation-specific antibodies to be used as diagnosis and therapeutic tools. Doing so, they identified 1 antibody specific to the FSH18, so the lighter form of the FSH, and this antibody is called ABX. So that's when we entered the program. They wanted to identify the epitope, the precise epitope of this antibody on the FSH. So we used our tool MAbTope, which is an epitope mapping tool, and MAbTope is based on docking, meaning that it needs 2 3D structures to dock on each other. So we needed to build these models. So for the antibody side, we just start with the VH and the VL sequences, and we can model the structure of the antibody using a modeling tool. On the antigen side, we had a crystal structure for the FSH, which reference is indicated here. But actually, if we didn't have the crystal structure, we could have built a model as well. So our methods generate millions of docking poses of the complex, and our artificial intelligence and machine learning tools apply here during the scoring of these millions of sequencing, and it will allow to identify the 30 best docking conformation for the complex. So it's what you can see here on the plot. Here you have the FSH alpha and beta, alpha in light gray and beta in dark gray. And you have here in colored ribbon the 30 best docking poses of the antibody. So from these 30 best docking poses, we are able to draw a heat map. So here it's the evaluation of the frequency of each residue to belong to the interface. So in purple here you have the high frequency residues, and after going in the lighter shades of blue, it's the less frequency residues. But still, these low frequency are still significant. We are so this is on the structure, and we can plot these residues on the sequence, as you can see here. So on the top here, you have the alpha sequence, beta sequence here, and we draw regions, interacting regions, covering the high frequency residues. So here we have one region, for example, covering these two high frequency residues, and here another one. These regions are the ones that are going to be targeted for experimental validation. So one thing that is important is that this whole process takes only minutes of computing, and if we compare that with experimental methods, of course it's way faster. These are the four classical way of doing epitope mapping, and the gold standard being the crystallography, which takes at least 6 months in the best case scenario. With MAbTope, so it's only minutes of computation, and to obtain the results in the SAS interface, and we need 2 weeks if we have a report to write, so meaning generate the figures and all the explanations that goes along. If we compare MAbTope to other docking methods, we are better because our algorithm gathered the 30 best docking poses on one side of the target, and actually this is around the actual epitope, which is shown here for the same target from the crystal. With other docking poses, actually the 30 best conformation are spread around the target, as you can see here. So we optimized protocols in order to go from the predictive data to experimental data, and to help biologists to use our predictions in their own lab. So the process of the validation is based on mutation introduction in the full-length target, and we are going to introduce mutations within each of the regions, meaning that in the end, what the biologists have to try in the lab, it's only the wild type to have the control, and after 4 constructs, 4 different mutants. Importantly, we only mutate the residues which lateral chains are exposed to the solvents, and we do not mutate the prolines. So the purpose of that is to ensure that the target will be correctly folded. So we introduce this mutation in the full-length target, and we overexpress the target in HEK cells, and after we measure the binding of the antibody to each of the constructs with cytometry. And the purpose is to see if we have loss of binding on one or the other of the regions, mutated regions. It means that we mutated the actual epitope. So for the validation in our case study of the FSH, actually we first had to build the wild-type FSH construct on which we could assess the binding of the antibody. And this expression was very tricky, actually, for several reasons. First, because the ABX binds specifically to FSH80, which is not glycosylating on the beta N7 residue. So in order to avoid the glycosylation of this residue, we introduced N7Q mutation. Also, FSH is dimeric, and actually we predicted that the epitope was over the alpha and the beta subunit. So we needed to create a fusion to ensure that the two subunits were going to be expressed as stoichiometric levels in the cells. And lastly, FSH is a hormone that is secreted and it's glycosylated, so it needs to route through the secretion pathway to acquire this glycosylation. So we needed to keep the membrane addressing peptide at the N terminus. But in order to retain the FSH on the cell membrane and not to be released in the media, we fused to the transmembrane domain of the CD8 here. So all these modifications of the genes were made in silico, and the construct was very easily obtained using twist technology. So first we assess the binding of the antibody on both of these constructs. So I forgot to tell you, we made two constructs, one fusion with the alpha and then the beta subunit, and a second one with the beta and then the alpha subunit. So we measure the binding of the antibody in APC, so here, and this APC signal is plotted on the x-axis. And also, we measure the overexpression of the target using a PE-coupled antibody targeting a flag. And this PE signal is plotted on the y-axis. So here you can see that for both of the cells, we have a deviation up and right, meaning that we have the target and it binds the antibody. So actually, the binding of the antibody was better for the beta-alpha fusion here. So it's the one that was selected after for the introductions of the mutations. So we introduced these mutations in the target, and it was very easy against using twist technology. And here are the mutations represented on the structure of the target. So each group of color means a mutation in one of the regions. And you can see that we have a very limited number of mutated residues. So we overexpressed the target in the cell and did our cytometry protocol, and we could measure the percent of the APC plus cells among the PE plus population, so meaning that the number of cells that actually bind the antibody among the cells that overexpressed the target. And you can see here that we had a significant decrease of this amount with the P1 mutant. And we also measured the mean APC signal in the different constructs. Here as well, we had a decrease for the P1M and also a decrease for the P4M. So what we can deduce from these results is that obviously the P1M and P4M include mutated epitope residues. So if we go back to the linear sequence at the beginning, we predicted 4 regions. 2 were validated experimentally, the P1 and the P4. Interestingly, the P4 is half of the region 4 is actually hidden under the security belt, which is this part of the FSH beta here. So we can conclude that the epitope of our antibody ABX is located over the region 1 and half of the region 4. So as a conclusion, I hope that here I convinced you that Mapsilico methods are fast and they are easy because they require very few information at the beginning. And just a few numbers now. Since 2017, actually, we've been able to work on more than 1,700 individual antibodies, targeting more than 55 targets in 9 different therapeutic areas, and we're coming from 40 customers. Among the 1,700 antibodies that we tried, we were able to compare our prediction with actual biological data for more than almost 600 of them. And we were able to evaluate that our predictions were correct in 85% of the cases. Most of our tools, as I told you, deal with antibodies, but actually we were also able to optimize peptides in order to develop a vaccine in the COVID-19 program led by a biotech company that we work with. And actually this vaccine entered clinic in 2020. Thank you very much for listening to me, and I hope that you have many questions.